Good morning, everybody. Myself, T. Mary Mrudula, working as a guest faculty for subject Microbiology in Tribal Welfare Degree College, Janagaon. Today, we will look into the topic of the present issue. So, all we know very well, the present issue in the society is the coronavirus. So, why this coronavirus is spreading like that? Before going to know that, we have to know first of all what is the virus. Means coronavirus means it is one of the species. In that, majorly it will comes under the family virus. So here, first we have to know what do you mean by the virus and what is its structure, what is its characteristics, and how it was discovered by the scientists, and what is the classification, and what are the tools we can use to study this particular structure of the virus, and what are the uses of virus, and the brief history of the coronavirus, as well as the uh, whenever any viral disease is attacked, we are going to recover from that disease by taking the vaccine. So we will know about that vaccines are also what are these vaccines, and what is the mechanism, or what is its importance. Okay, let's come to the point that first of all, what do you mean by the virus? So here, viruses are the particles. Here we won't call these viruses as they are the microorganism. Okay, it comes under the microorganism, but we won't say it as a living organism. Directly we won't say it as a living organism. Why? Because when it is outside of the body, it is going to act as a non-living particle, or it is going to as an act as an agent. So that there is an whenever also we are going to define the virus as like this, that it is an infective agent or infective particle that is called as a virus. So generally these particular virus will contains majorly three parts. One is protein and nucleic acids, as well as the envelope. These are the mainly three parts which are helpful for that particular virus in their multiplication or in its reproduction. Generally we won't call here for virus also as in a reproduction method. Why? Because it is not going to show any sexual mode of reproduction or any asexual mode of reproduction. It is going to multiplication. So there is lot of difference between the multiplication and addition. So addition means like it's reproduction. Multiplication, we know it, the number will be doubles. Addition means just added one more one, but here it is going to multiply. So that the reason only actually the virus is very dangerous when you compare with the other one. Is it clear? And when coming to the introductory part, majorly here it is content, as I told just before, that it is having mainly the three parts, that is envelope, genetic material, as well as the protein coat. And the shape of this particular virus will be several types: helical, icosahedral, as well as the complex structure. Means structures we can't explain that much of clearly, that as it is having a complexity in its structure. So generally we know the meaning of helical, icosahedral, complex. I will show the structures how they will be, and helical means how it will be, icosahedral means how it will be, a complex structure means how it will be. And coming to the size, the size of this particular virus will be ranges from 20 to 300 nanometers. 20 to 300 nanometers. Why? Because when you compare with other microbes like bacteria and algae and fungi, it will be smaller than, very smaller than those. Is it clear? And here, after completion of the introduction, when we have seen what is its basic structures, means it is having a capsid. This capsid is going to play the major role in multiplication of this particular virus. And here, whenever we are going to see, this virus will not divide. As I told just before, that it is not going to undergo any sexual or asexual reproduction. It will simply multiplies in host cell with the help of a vector. Without the vector, this particular virus will not infect any host. Clear? And these particular viruses are evolved from the plasmids in between the cells. Till now, 5,000 species of viruses have been discovered by the scientists. Okay, 5,000 species are discovered by the scientists. So, what are those species? Means which are important species only we will see today. Is it clear? And coming to the discovery, here this discovery of the viruses is done by the many scientists like Chamberlain as well as Ivanovsky, Bejernik. So first, if you have seen, in 1884, a scientist called Charles Chamberlain, he invented a filter. Means here filter is a sterilization technique of the viruses. Means if you want to sterilize the viruses, we have to use the method called filtration method. So which filters he has used means the Chamberlain Pasteur filters. These Chamberlain Pasteur filters are very useful in order to filtrate the or in order to sterilize the viruses. Like that, actually he found that okay, these particular particles which left in this particular filter are the virus. Like that, the Chamberlain was invented this particular viruses. Coming to the Ivanovsky also used the same filters, and he has filtered the tobacco mosaic virus from the tobacco plant. When he has observed some infected leaves, he has collected that leaves and he has crushed that leaves. From that leaves, he has filtered by using the Chamberlain filters, and then he has separated the virus. Like that, in that way, the plant viruses was discovered by the Ivanovsky. Coming to the Bejernik, actually Bejernik was also tried to know how the division of this particular viruses is going on, but he was failed in that particular. Later on, a scientist called Ttwrdc, he is an actually bacteriologist, not virologist, but he has done the work on the viruses. He came to know that some of the viruses are going to infect the bacteria. Those are nothing but the bacteriophages. Bacteriophages means viruses are not only infecting to the plant, not only infecting to the animals, they are infects to the bacteria also. Generally, when you observe other microorganisms like your bacteria and fungus, anything, one of the microorganism is not going to attack another microorganism. But speciality of this viruses is that virus is also a microorganism, bacteria is also microorganism, but small variation is that size varies. So certain variation is also going to affect. Means here virus is also a microorganism, bacteria is also microorganism also. This particular virus is going to affect on the bacteria. That was discovered by a scientist, Frederick Ttwrdc, in the early 20th century. And lastly, the clear picture of this particular virus was known by using the electron microscope only. This electron microscope discovery was done by Knoll and Ruska in the year 1931. First images of this viruses was taken with the help of this particular electron microscope. Means by using light microscope we can see the structure, but we can't take the image. That image we have taken with the help of this particular electron microscope. Like that, in the year 1931 we got the images of this particular viruses, and in the year 1935 the biochemist, not virologist again, biochemist Wendell as well as the Stanley, these two have examined the tobacco mosaic virus, and what they found means these particular are made up of mainly from the protein. So as I told, when we observe the structure of a virus, it is having a protein coat ani. So how we came to know means due to the work of this particular Stanley as well as the Wendell, they have said that these particular viruses are made with the proteins, and as well as lastly this 1931 one more invention was also done regarding the viruses, that is the influenza. as well as the this influenza, they have fertilized in the chick eggs. Who fertilized means Ernest William as well as the Good Pastures are the two scientists. They are the pathologists. They are the pathologists. They have grown this particular influenza is a virus in the chick of the chick eggs. In that they have inoculated and they have told how they are going to infect the organism. Is it clear? And coming to the spread as well as what do you mean by the vector. So here when we have seen how these are going to spread means here these viruses are going to spread in many ways. Many ways means see here they transmit or they spread this disease to the animal, they spread this disease to the plants, they spread this disease to the human beings, they may spread to the disease to the even bacteria. Just now I told means it is going to attack on the bacteria also. Okay, to anyone it want to attack also, it need a vector. It need a vector. So what do you mean by the vector means here vector means it is a living organism which is transmits the infectious agents, which will transmits the infectious agent. Means simply the vector is a mediator between the virus as well as the host. Means when this vector carries this particular virus into the host, then only the multiplication will start. Until and unless this vector is not there means there is no virus multiplication. Is it clear? So here this virus will wait for the suitable vector. Generally what are the vectors this particular virus is going to use means like your houseflies, mosquitoes as well as the ticks. Okay, houseflies, mosquitoes, ticks, etc. These they are going to use, these living organisms they are going to use as an vectors. With the help of that vectors they are going to transmit the disease to the animal or the plant or the what we'll call bacteriophages also even sometimes. And here so we have seen the spreading. So here means plants and animals and bacteria etc. for all there it is going to spread I told. Not only that here it is going to transmitted by the fecal oral route also. Means oral means when means through our mouth also the virus can be entered, that as a contamination of hands, food and water, and in such a way also it is going to spread. Is it clear? And another more, suppose example if you have taken HIV, this HIV will be transmitted by the body fluids. Okay, body fluids, through that this particular HIV is going to be spread. Suppose dengue virus, it is going to spread by blood sucking insects like your mosquito. So your mosquito will carry the blood and will cause the disease. Rotavirus, this rotavirus will be caused due from the infected children to the other infected children. Suppose if I got this rotavirus means nothing but which is diarrheal disease, actually it is diarrheal disease, rotavirus is this. So if some other person also came in contact with me, the hands which I used, that hands if I touch it to that other person, he also get that particular disease. So in that way also this virus is going to spread. And here no antibiotics can be used to treat the viral disease. Only antiviral drugs as well as the vaccines are only the solution to treat the diseases which are caused by the viruses. Anyhow we are going to see at the end of the topic how these vaccines are going to work on the viral diseases. Next, coming to the characters means why these particular viruses are that much of widespread when compared with other bacteria and other microorganisms. Means here, see here it is an obligate intracellular parasite. Obligate intracellular, not intercellular also, intracellular. Okay, in the cell, not in between the cell, inside of the cell it is going to do. Intra means inside, inter means in between two. Here in between two it is not happening, it is happening directly inside of the cell, that too very obligatively, means very complicated, obligate. I have a lot of obligations with that person, generally we will use that term. No, obligations means always the complications only will be there with that particular. Means that much of effective, that much of infective character it is. So to whom it is going to means for bacteria, for protozoan, for fungi, algae, plants and animals, not leaving anyone, for everyone it is going to cause a matter. So that is one of the character is obligate intracellular parasite, and as well as it is an ultra microscopic. Microscopic is that can see only with the help of microscope. Ultra means smaller than that. So that the reason only with the help of electron microscope only we can see the clear picture of this particular virus. And already we have seen what is its range. As well as coming to the next part, it have an this structure whatever when we observe, the structure is very complex structure or compact structure it is having, and as well as it do not independently fulfill its characteristics of life. Okay, independently fulfill అంటే తన సొంతంగా తాను ఫుల్ఫిల్ చేసుకోదన్నమాట తన లైఫ్కి సంబంధించి. వై ఇట్ వితౌట్ ద వెక్టార్ ఇట్ కెనాట్ fulfill its life. Simply thing. Without any help it can't fulfill. Okay. Means that particular vectors are not there means anyhow they don't have the life. As well as they are acting as an inactive macromolecules outside of the host cell. Means when they are outside they are acting like an macromolecule only. Again it is not acting as a microorganism. It is macro and as well as inactive. So that the reason we are not going to take care about that. But once it found the vector means when it is enters into the body, it will start its multiplication, not addition also. Is it clear? That is the speciality. Here it has a speciality that whatever the synthesis of this genetic material, whatever the genetic material present in the virus, that genetic material synthesis will be occurs within the host only. Without that host there is no synthesis of the genetic material. So here means what is the genetic material of the virus we will see later. Means here DNA will be there, RNA both will be there. Again in that also two separate means based on the classification we will see what types of genetic material a virus will be having. And here virus will not have any enzymes and it have lacking of the machinery for the synthesizing of the proteins. Okay, whatever the viral infections will be there, they will be mild to life threatening means acute to chronic will be there. And next compare the difference between a virus as well as a cell. Here what is the main difference between a virus as well as a cell means here virus is having genetic material whereas cell is also having a genetic material, but here the multiplication will be occurs within a host cell only, but the cell will can divide itself means asexual or sexual method. By using sexual method or asexual method the cell can divide, but whereas the virus cannot divide likewise, but both are having the genetic material. Virus will have DNA and RNA both the genetic materials, but whereas the cell have only the DNA materials. And here when we have seen energy usage, it will your virus will not obtain any energy, but whereas cell will obtain its energy. And response to the environment, virus cannot respond to the environment, but the cell will respond easily to the environment. Okay, and change over on time. Okay, both virus and cell both can be changed over the time. Okay, time being there is lot many changes can be seen. See here, see the structure of an virus as well as the cell. Here you can see here when you compare these two figures, the cell is having the particular structures. We can divide them: nucleus, endoplasmic reticulum, like Golgi apparatus very clearly. But here it is having very complex structures. It is very compact. Cell is very freely it was there, but your virus structure is very compact, and there is no cell membrane for the viral structure, whereas your cell is having a cell membrane. Is it clear for you? Now next, maybe you may get some doubts that whether these structure will be rigid, whether it is a big or very flexible or not. So here anyhow virus structure is not big as well as also not flexible. Okay, and here if this structure, whatever the structure of the virus will be there, how it should be means, it should provide the protection for the genome, as well as it should allow the virus to move from host to the next, as well as it should have the capacity to attach to a new host. Why? Because if attachment is not done means there is no multiplication. Is it clear? And next, coming to the size, as we have seen before itself only. So here if we have seen there the virus range means 20 nanometers to 400 nanometers it will be there. But here is one word is there, virion size ranges from 10 to 400. Anyhow both are same, but small variation will be there. We will see what is the main variation between a virion as well as the virus means. Here see, virion means fully formed virus. Fully formed virus means which is having the protein coat, envelope as well as the genetic material. Okay, the entire virus particle total is called as virion. Is it clear? Means which is having the capsid also in it. Means the nucleic acids are closed by a structure called capsid. See here you can see whatever the black threads like structure is there, it is actually the nucleic acids, means genetic material. Either DNA will be there, or either RNA may be. That is closed by this particular capsid. These capsids are made by the structures called capsomeres. And basic components of this virus will be 3 nucleic acids, capsid as well as the envelope. Okay, fine. Coming to the classification. Here the classification of the viruses mainly done based on the several factors. What are those several factors means? First one is based on the host or based on the habitat. Okay, on which host it is going to do its multiplication. Based on that classification was done. Based on the structure the classification done. Based on the chemical composition. Based on the genetic makeup, similarities on the genetic makeup, as well as based on the some other characters, that is genetic material presence also. Okay, what that we will see. Okay, here so likewise based on all these factors, nearly 63 families and 263 genera, 3 orders were there in the virus. So in this classification, see, based on the host, based on the habitat, how many? 3. We already discussed also, but it will come under the classification. Animal viruses are there, plant viruses are there, bacterial viruses. Okay, based on the host or based on the habitat, 3 types of viruses. One is animal viruses, plant viruses as well as the bacterial viruses. Is it clear? If you have seen some examples, animal viruses, your rabies, polio, mumps, chickenpox, smallpox as well as the influenza. Plant virus is your tobacco mosaic virus, banana streak virus, carrot thin leaf virus. Bacterial viruses will be the bacteriophages. And next coming to the based on the genetic makeup or based on the presence of the genetic material. So as just before we discussed, DNA and RNA both are going to act as a genetic material. Again in that DNA and RNA also, so first we will see the DNA viruses. See, DNA viruses are also enveloped will be there, non-enveloped will be there. Enveloped will be there, non-enveloped will be there. So what is enveloped and what is non-enveloped? I will show a pic to you, then you will understand easily what is enveloped and non-enveloped. So DNA viruses are also enveloped as well as non-enveloped. Again in the enveloped one, double-stranded genome. Examples for the enveloped double-stranded DNA is pox virus as well as the herpes virus. Non-enveloped double-stranded DNA is adenovirus and papoviruses. Non-enveloped single-stranded example is parvoviruses. RNA viruses. So again RNA also enveloped will be there, non-enveloped will be there. Single stranded will be there, double stranded will be there. So here single stranded as well as double stranded will be there. What are the examples of the single? Means here arthro, mixo, viridae as well as adenoviruses as well as paramyxoviruses, rhabdoviruses, flavivirus as well as your coronavirus is also here is their example. So here is coronavirus will comes under which one? Means RNA viruses enveloped as well as a single stranded genome or single stranded gene will be there, not a double stranded one. And the non-enveloped single stranded is your picornavirus as well as calcivirus, whereas the double stranded genome virus example is your reoviruses. See here, envelope, non-envelope means how it will be. Naked, I mean this envelope structure will be not there on the top. Here you can see a purple color structure will be there. That envelope is absent in the non-enveloped viruses. So this envelope is there means those are called as an enveloped viruses. Envelope means whatever the genetic material and that capsid will be there, that is surrounded by again one more layer. That is called as an enveloped virus. Okay, we can call in another one naked viruses are also. Naked means which don't have the envelope. Certain viruses are called as naked viruses. Is it clear? This is the classification of the viruses. Next, coming to the tools can be used for the studying of the structure. Means now till now I am telling that the virus is having this protein coat as well as the genetic material, nucleocapsid, envelope will be there, and it is having some glycoproteins and it is having spikes. How I came to know all these things means here our scientists already have done the experiments on this structure and they revealed this structure. So how they have revealed these structures means they have used some tools to reveal the structure of the viruses, and they have confirmed that which are the single stranded RNA viruses, double stranded RNA viruses, single stranded DNA viruses, single stranded RNA viruses, plant viruses, etc. things they have revealed. What are those tools means? Mainly the tools mainly used is three. One is electron microscopy, and another method is X-ray diffraction method, and the third one is the cryo-electron microscopy. Cryo-electron microscopy. So these are the three tools they have used to reveal the structure of the viruses. Next, see here you can compare the structure of a virus with a bacteria. You are seeing here, see a bacterial structure is there. When you have seen a bacteria structure, and this remaining all are the virus structures actually. How small they are when compared with the bacterial structure, see here itself only. How small? How small? Means one bacterial cell is not equal to these all viral structures. Okay, all that much of differences will be there. Next, coming to the uses of virus. Oh, you may feel, will we have the uses with the viruses? You may get the doubt. Why? Because first of all, whenever human beings, any people, if you say word microorganism, if you use the word microorganism means only, they will think that all are harmful only, but it is wrong. Microorganisms useful also is there. Okay, virus, okay, maybe bacteria will be useful means we may believe. How virus will be useful, you may get the doubt. Okay, how they are useful? Okay, let me explain. See here, we are taking so many vaccines. Okay, so many diseases like your polio as well as measles, mumps. How means you are nothing but by using of this particular viruses only. Why? Because with the help of these viruses, the scientists have developing the vaccines. Already the first development of this vaccine was done by the scientist Edward Jenner for the disease smallpox in the year 1700s. At that time only there is a history behind it, that here when a milkmaid person was came to the Jenner with a small blisters on his skin, he has taken the pustule from that and then he has injected to the infected person, then the person was relieved from the disease. Like that Actually, he means he has done the research on that, then finally has invented the vaccine. Means with the help of virus only we can prepare the vaccines. Okay, that is one of the major use with the virus. So first started for smallpox, later on came for other diseases are also, like your, see here, human diseases, wart disease, common cold, influenza, smallpox, Ebola, herpes, AIDS, chickenpox, rabies, and etc. For all these now for all these human diseases we have the vaccine which can be prepared only with the help of viruses. One thing you have to remember that whatever the vaccines we are preparing with the help of these viruses, we can prevent but not treated. Okay, but not means complete treatment was done. But here when you have compared with the antibiotics, here there is no antibiotic can cure the viral diseases. Vaccines will be most effective. They are the most effective one. Is it clear? These are the uses. Coming to the coronavirus, we'll see very brief history of this corona, just 5 minutes. So here we know now present issue is the coronavirus. So this coronavirus is a large family virus only, which is causing the diseases to the human beings, animals, as well as the bats, camels, as well as the quails. So here now this whatever the coronavirus which is caused to the human beings, this particular coronavirus will belongs to which family? Means Coronaviridae family of Ttwrdc order. Ttwrdc order, Coronaviridae family. From that family this particular coronavirus was came, and other one also. This particular coronavirus can cause the disease SARS, that is severe acute respiratory syndrome. That's why see here nowadays we are hearing this news that the person who are attacking and who was getting the breath problem. Breath problem is nothing but inhalation problems are going to cause. Here before only we know this SARS means this coronavirus generally will affect on the respiratory system only, on the respiratory system only. So whatever the coronavirus which is caused to the human beings is SARS-CoV-2. SARS-CoV-2 species, means SARS-CoV-2 is species. One is given to another one. Means here in some colleges or some we have some numbers, no. Likewise here they are also have numbering that to differentiate that which is causing to the human beings and which is causing to the animals. Here that which is caused to the human beings is SARS-CoV-2 strain. It is a new strain. Okay, which is not identified previously in the human beings, but now it was identified. So how would this coronavirus, human coronavirus were origined? Origined means it is we don't know exactly, but there is a belief that here these coronavirus were origined from the bats, and from there too it was jumped to the camels, then to the human beings, means like that it was came to the human beings. Now we are believing actually. Okay, so here like that now which was identified for the human coronavirus is which one? SARS-CoV-2. If you see the structure of this coronavirus, how it will be means it is having a special structure, means general virus how it will be like that only it is also having. It will be showing some crown like spikes on it. See, you can see crown like spikes here, you can see these crown like spikes are nothing but made up of protein, and this outer whatever crown like structure will be there, due to that only it was named as an corona, means crown like spikes are present, no. Likewise that's what the reason actually name was given as coronavirus. This coronavirus is causing a disease called COVID. Okay, COVID is also not a virus actually. Again, COVID is a disease. Corona virus is a virus name. Is it clear? And what is the size of this corona is 16.5 to 125 nanometers. And it is having already, just before we have seen, so it is in a single stranded RNA genetic material. It is having the single stranded RNA genetic material, and this genetic material size will be 26 to 36 kbs in length. Kbs in length. Is it clear? Next, coming to how to come out of from this viral diseases. Not only all viral, now whatever present corona is there, for this also doctors are suggesting us to take the vaccines. Why? What is the importance of this particular vaccines in the disease or in this particular coronavirus disease? Means here vaccine have some capacities. What is that capacities? We will see just briefly. Again we will see. So first we will see what do you mean by the vaccine. Here vaccine will be given to improve your immunity. To improve your immunity. So your body to develop the immunity means you need to take these particular vaccines. Why? Because when we have taken these vaccines, they are going to increase the production of the antibodies inside our body. That antibodies are going to find with the antigens which have entered into your body. Means antigen is nothing but your foreign particle, means your coronavirus which is entered into your body is an antigen. With that antigen, someone has to come to fight. Those are nothing but antibodies. So that improvement means that antibody production was improved, means nothing but your immunity was improved. To improve that immunity, we have to take this vaccine. Okay, to take the vaccine. So here what is it means? It is vaccine is a biological preparation which will helps in the improvement of the immunity to a particular disease. Suppose if you have taken means to understand you how biological will be prepared means different types are there: live attenuated, killed attenuated, inactivated. Means the particular foreign particle antigen which is there, we are going to inactivate that virus, and after inactivating we are injecting into the body. Then what will happen here? Your antibodies will think that they are the real active virus, and then they will try to kill that particular virus. Like that what will happen? There is an improvement in the antibody production. Like that when your antibody production was increased, what will happen? When the active virus or active foreign particle entered also, it will respond same. When you have given inactivated, how it will respond? At the time of active also it is going to respond same way. By that we can protect ourselves from that particular disease. So we can protect ourselves from like this by taking the vaccine. Who told to us? Means here again your Edward Jenner first by discovering for the smallpox. At that time only he has told the what is the principle involved in the vaccine preparation. The same principle only till now we are using for the preparation of the vaccines. Is it clear? So here this vaccine is a word came from the Latin vacca. Vacca means cow. Vacca means cow. Means first actually from cow pustule he has taken Edward Jenner. So here based on that only actually that name was continuing. Latin word vacca means cow. From that we are using this one. And after Edward Jenner in the year 1885, Louis Pasteur as well as the Emil Rox, these two also have developed the vaccine against the rabies. And after smallpox, the next vaccine came is rabies. Later on the other rabies have came. So simply a overall look of this particular mechanism of the how vaccine is going to be worked means, see here when you closely observe this picture, see here you are seeing a foreign microbe with an antigen. When it is enters or when it attacks on the cell is going to be engulfed this and it is going to be processed. Then what will happen here? It is going to distribute to all the cells, and means here memory cells will be there, suppressor cells will be there, helper cells. Means help some cells will be there to help, some cells will be there to memorize. Means the particular it is a foreign particle. Okay, if it is came next time we have to kill. That has to be remembered by a cell called memory cell. Memory cells will helper cells will help to kill that particular foreign particle. Why? Because in our body also, only one cell cannot improve your immunity. All the cells has to be unite. You know one proverb, when we will be unite, there will be a more strength. Same the sense, all the cells when they are together, they will be having more strength, they can distribute the work and they can attack on that particular foreign particle or on the antigen and can improve your immunity. Is it clear? This is the mechanism. Next, coming to the importance. What is the importance of this particular vaccines? Here, according to the NPH, it is a cornerstone for the public life. Why? Because here these vaccines can be given to the infants, children, adolescents, adults. Okay, there is no particular age differences. All can be taken. But here depends upon the dose. That's the dose is going to be varied from the infant to children, children to adults. And for health workers, for military persons, prisoners, as well as CSWs, for all these, I think all these you are seeing nowadays, who are continuously communicating or who are continuously touch with the public, who are serving the people like in the hospitals, they require these vaccines must and should. Why? Because they have to treat the patients, means first of all they have to take. And as well as it is now, means the vaccines whatever we are developing, that now it is in a developing field. It is developing field. So here we know one thing is that public health is also one of the major criteria for the government. So that the reason here, along with the food and home shelter etc. things and providing water, here giving of the vaccines is also very important. So that the reason, see nowadays for the children till 14 years of age, they are taking the vaccine. Why? Because till the 14 years of age vaccines are there. Whatever those vaccines are there, every vaccine is given by the government freely only. Means that is the importance of this particular vaccine in order to develop your immunity. Is it clear? And as well as whatever the vaccine we are preparing, compulsory it should be cheaper. Suppose anything we want to purchase, means it should be able to purchased by even low poverty people also. Like that only it should be prepared. And the vaccine whatever will be there nowadays, not only to treat the disease, vaccines will be is given to improve the vitamin and mineral supplementation for the bed nets also. For the bed nets, means we are giving combination with that means it is going to cure the disease as well as giving of the supplements of these particular vitamins as well as the minerals also. Is it clear? I hope everyone is understood this one. So thank you for everyone listening patiently, and thank you for giving me this opportunity to our